Welcome back to the Victory Education Lounge. I'm your host Maurice, and today we're diving into the microscopic world of molecular mayhem, the science behind sickle cell, Part 5. Before we embark on this molecular adventure, please take a moment to like this video, subscribe to our channel and share your thoughts or experiences in the comments below. Your engagement helps us reach more curious minds with this fascinating information. Imagine your body as a vast, intricate factory, with proteins as the tireless workers keeping everything running smoothly. Now picture one of these workers suddenly changing shape, causing a chain reaction of chaos throughout the entire system. This is essentially what happens in sickle cell disease at the molecular level. Ever wondered what it would be like if a tiny mistake in your body's blueprint could cause a chain reaction of chaos? Well, buckle up, because we're about to dive into the intricate world of molecular mayhem, where just such a scenario unfolds in the case of sickle cell disease. So picture this: your body, a sprawling complex factory, and proteins, the tireless workers keeping everything humming along. But what happens when one of these workers changes shape? Suddenly there's chaos, a domino effect that sends ripples of disorder throughout the entire system. This is no mere abstract concept, this is the reality for people living with sickle cell disease. It all starts with a tiny error in the genetic blueprint, a mutation in the HBB gene. This gene is like the instruction manual for making beta-globin, a crucial part of hemoglobin, which is vital for carrying oxygen throughout our body. This genetic mutation, however, leads to the production of a different kind of hemoglobin, known as hemoglobin S, or HbS. And the difference between normal hemoglobin and hemoglobin S? It's just a single amino acid in the beta-globin chain out of 147. Once this hemoglobin S lets go of its oxygen, it reveals a sticky patch on its surface. This allows the hemoglobin molecules to stick together, forming long, rigid fibers. These fibers push against the cell membrane of the red blood cells, morphing them from flexible discs into rigid sickle shapes. And that's when the real trouble begins. These misshapen cells can stick to the walls of the blood vessels and to each other, causing blockages that can lead to pain, organ damage, and a host of other complications. So there you have it. A tiny mistake in the genetic blueprint leads to a chain reaction of molecular mayhem. Imagine this: just one amino acid out of 147 in the beta-globin chain goes awry, and it's enough to kickstart a molecular mayhem. Now, let's see how this tiny mistake can lead to a domino effect of complications. Imagine a line of dominoes standing perfectly still, each representing a step in the process of producing and using hemoglobin in our bodies. A single nudge at the beginning of the line, as insignificant as it may seem, can set off a chain reaction that topples every domino in its path. In the case of sickle cell disease, this nudge is a single genetic error in the HBB gene. This minute change leads to the production of abnormal hemoglobin, the protein that carries oxygen in our red blood cells. Instead of the normal smooth hemoglobin, we have hemoglobin S, a sticky, rigid version that behaves quite differently. When hemoglobin S releases its oxygen load, it reveals a sticky patch that allows it to stick to other hemoglobin S molecules. Like dominoes falling into place, these molecules form long, rigid fibers. These fibers in turn push against the cell membrane, transforming the flexible, disc-shaped red blood cell into a rigid sickle shape. This is our first domino falling, setting off a chain reaction. The misshapen cells, like fallen dominoes, can't move smoothly through the blood vessels. They stick to the blood vessel walls and to each other, creating blockages. These blockages prevent the normal flow of oxygen-rich blood to the body's tissues and organs, leading to pain, organ damage, and a host of other complications. Each fallen domino represents another step in this chain of events, another complication arising from this single genetic error. So, a single error in the genetic blueprint can lead to widespread chaos in the body's system. Just like a line of dominoes, a single nudge can set off a chain reaction that brings the entire system crashing down. That's the domino effect of sickle cell disease, a powerful illustration of how a tiny mistake can lead to significant consequences. Scene script: how does this molecular mayhem play out in real life? Let's explore some scenarios. Our first scenario is about Sarah, an ambitious marathon runner. Sarah has sickle cell trait, and during a particularly grueling race she collapses. The intense exertion and dehydration have triggered a sickling event in her red blood cells, causing her to experience a sickle cell crisis. Next we have John, an adventurous mountain climber. While on a high-altitude hike he suddenly falls ill. The lower oxygen levels at such heights have increased the formation of hemoglobin S fibers, leading to a painful sickle cell crisis. Our third scenario involves Maria, a scuba diver who loves to explore the deep blue sea. Maria is unaware that she carries the sickle cell trait. After a deep dive, she experiences intense joint pain. The pressure changes associated with deep-sea diving have induced a sickling event in her red blood cells. Then there's Jamal, who braved a polar plunge challenge. The sudden drop in temperature as he plunged into icy waters triggered rapid sickling of his red blood cells, leading to a painful crisis. Lastly, we have Aisha, a diligent student facing the stress of exams. The heightened stress hormones in her body have triggered an increase in sickling, resulting in a sickle cell crisis. These scenarios illustrate how various environmental factors can interact with the molecular abnormalities of sickle cell disease, leading to crises. From physical exertion and altitude to pressure changes, temperature drops, and even emotional stress, many factors can trigger the sickling process in people with sickle cell disease or trait. These examples demonstrate the complex interaction between our bodies and our environment, and how a microscopic change can have macroscopic effects. Understanding these interactions can help us better manage sickle cell disease and potentially prevent crises, improving the quality of life for those affected. So, stick around as we delve into potential solutions, management strategies, and prevention efforts in the next part of our series. Until then, keep exploring, keep learning, and remember, even in the face of molecular mayhem, knowledge is power. Is there a way to manage this molecular chaos? Let's explore some solutions. First on our list is a medication called hydroxyurea. This pharmaceutical marvel works by increasing the production of fetal hemoglobin in the body. Now, fetal hemoglobin is the good guy here because it doesn't participate in the sickling process. This means that more fetal hemoglobin equals less sickling, which in turn results in fewer pain crises and a reduced need for blood transfusions. Sounds pretty fantastic, right? Well, it's not all sunshine and rainbows. The use of hydroxyurea requires regular monitoring to ensure it's doing its job without causing harm. Additionally, potential side effects include a decrease in fertility, which can be a significant concern for some individuals. Next up, we have L-glutamine. This is an amino acid that plays a crucial role in protecting red blood cells against oxidative stress. In simpler terms, it's like a bodyguard for your red blood cells, shielding them from harm. This protection can result in reduced pain crises, and the best part? It's available as an oral medication, making it an easy addition to daily routines. However, just like hydroxyurea, L-glutamine isn't without its drawbacks. It can cause nausea and headaches, and the long-term effects of its use aren't fully known. So, while it can provide relief, it's essential to consider these potential downsides. While these solutions offer hope, they also come with their own set of challenges. Continuing our exploration of potential solutions, let's discuss a few more options. First up, we have crizanlizumab. This is a monoclonal antibody that blocks a protein called P-selectin, which is involved in the sticking of sickle cells to blood vessel walls. The benefits of crizanlizumab are that it reduces the frequency of pain crises and hospitalizations. However, the drawbacks include potential side effects like nausea, joint pain, and fever. Next, we have the promising field of gene therapy. Here, scientists aim to correct the genetic mistake that causes sickle cell disease right at its source. The idea is to use a modified virus to deliver a healthy version of the HBV gene into the patient's own bone marrow cells. This therapy has the potential to be a one-time treatment that could provide lifelong benefits. Nevertheless, gene therapy is still in its experimental stages. It's a complex, expensive procedure, with risks that include immune reactions and the possibility of the virus causing other genetic changes. As we delve into these potential solutions, it's crucial to remember that each person with sickle cell disease is unique. Factors such as the severity of the disease, the presence of other health conditions, and the individual's lifestyle and personal preferences all play a role in determining the best treatment approach. Therefore, personalized treatment plans, developed in close collaboration between patients and their healthcare providers, are the cornerstone of effective sickle cell disease management. In conclusion, sickle cell disease is a complex condition that requires a multifaceted approach to treatment and management. As we continue to unravel the mysteries of sickle cell disease and its molecular mayhem, new possibilities for treatment and management are emerging. In this unending quest for knowledge, every discovery brings us one step closer to victory. Before we wrap up, a quick disclaimer. The information in this video is for educational and entertainment purposes only. Any health-related decisions should be discussed with a licensed physician. Thank you for joining us in the Victory Education Lounge. Don't forget to like, subscribe, and share your thoughts below. Your engagement helps us create more valuable content and reach those who are curious about the intricate world of molecular biology. Until next time, this is Maurice, reminding you that understanding the molecular mayhem of sickle cell disease brings us one step closer to unraveling its mysteries and finding a cure.